What I present you today is an experimental talk in the line of what Vincent Hakim presented on the theoretical side when was that Saturday. So you see maybe a few common slides that he stole from me, and I stole from him. And besides that, there are other aspects to this story that I'd like to tell you today, plus a little extra if I have time. So, collective cell behaviors. I used to use this slide all the time. That was my typical introductory slide. And then I was tired of it, put it aside. And I put it back because of a remark that Hugues Chaté, who is not here today but gave a lecture Saturday, told me one day. So, of course, you recognize these animals, these starlings here, and fish there. I don't know what kind of fish that is, by the way. And there is a huge difference. As you say here, sky is the limit. There is no limit. You know, no boundaries. They can go wherever they want. But in the case of fish, it's a different story. Most of the pictures that you see in the literature, they're taken in aquariums. And of course, by definition, aquariums are not infinite. One of the most famous of these one, I don't know about that particular one, is taken at the Monterey Aquarium, and the Monterey Aquarium is a cylindrical fish tank, huge one. So if it's cylindrical, well, you've put goldfish in an aquarium already, I guess, or you've seen that. It's no wonder that, you know, they make swirls. It's collective, all right, but it's not, you know, really, you think of it differently. And that's the point I'd like to address today in the behavior, in the emergence of behavior in these multicellular systems, is the importance of boundary condition or lack of boundary conditions. So there are so the aspect of finite size, if you want. Why is it important? It's important for fish. If they're in a fish tank, obviously, you would, right? I would. It's important in vivo. In many cases, in embryonic development, in morphogenesis, you often deal very often with small populations of cells that have to migrate from one place to the other within constraints. It's not like an open system, not at all. They have a certain place where they need to go, their mission, if you want, and they need to do that together, but within very high constraints. So you need to take these into account. There are less desirable situations where you start exactly the same way. You start with a few cells that make a tumor in the case of cancer, and these cells are bound to be together. They're not that numerous, at least at the beginning, and before they evolve into something else, which will be single cells migrating away from this initial tumor, they have to deal with it at a small number within spatial constraints. So that's the point. But it's also maybe a less known aspect is the importance of this in biotechnologies. You've all heard of what is called cell chips, where you put a small population of cells within chambers, and you measure on this population of cells the phenotype they acquire when they're subjected to a certain drug or to genetic modification or whatever. This is from a company, by the way. Here there are gradients of whatever they want to put it. But the point is, you see the cells here. Again, it's what we would call a mesopopulation. It's not infinite, it's not single cells, it's in between. And obviously, well, not obviously, but what happened is that the behavior of these populations will also depend, as Vincent mentioned Saturday, that's all right. We'll also mention on the size of this population. So it's important in that respect too. And all the microfluidic technique that we use and are used more and more to assess the behavior of cells in these conditions. To deal with this aspect of cell-cell interaction, you have two aspects to consider. One of them is how two cells that migrate independently at first will interact, and how will that affect their respective velocities. And the other is how will that affect their degree of proliferation. if they come together and make a sort of a dense colony, then how will they move within this colony, and how will they continue or not to proliferate within that colony? And these two are called contact inhibition. It's a one wording only, although it covers two very different notions, locomotion and proliferation. Very shortly, I'm going to address these two aspects in this introduction. So we know more or less how a single cell migrates on the surface. It protrudes at the front end and retracts actively at the back end with the mobilization of myosin motors here. This is, of course, details are still to be worked out, but this is mostly known. Oops. Here it is. The question is, what happens when you have many cells? And a few cells, it's okay. We sort of know that. Two cells colliding one into the other will just stop there and then go back into opposite directions. That has been known for a long time. This is not that long ago, but already in the 50s, 60s, Michael Abercrombie really formalized that and used this term of contact inhibition of locomotion. That contact then redirects the cell in another direction. So this is sort of well known. However, if you look at a monolayer, where all cells are packed one with the other, they all interact together, so then they don't have any space where they can, as it says, redirect the cell. So what happened then? Well, what happened is completely different. It's a whole new ballpark. Cells are highly motile. This is widely accelerated, of course. But cells are very motile. That's the point. Contact inhibition describes well what happened with a couple of cells. Not that much what happened when you have a real population, a dense monolayer of cells. It's completely different. And we know how that works. Cells within the monolayer extend lamellipodia, protrusion, pretty much like here. But they can't do it on free space, so they have to go under the other cells, as it is represented here. So they extend the lamellipodium under the other cells, and they could use this mode of locomotion to migrate while other cells surround it. But of course, the net output of that is that in this picture, if you wait some time, the velocity on average will decrease and go back to very small values. And that's what we observe here. You have the average velocity here, the time, and you see that within, let's say, 10 hours, the velocity decreases by a very significant amount. Here you have only local motion left and not much. At the same time, the density increases. So what you can see already on that plot, and I'll come back in a minute to this aspect, is that the velocity really plummets once you reach confluence, which is this vertical line here, whereas the density doesn't seem to care that much, right? So the two notions are pretty much uncoupled. They go in opposite directions, but they don't follow the same exact trend. So that brings me to contact inhibition of proliferation. Again, it was postulated that normal cells, when they reach confluence, they would just stop dividing. That would be it. And that would be a sort of a hallmark of cancer. Transformed cells don't do that. They grow in the third dimension. But again, this is a very, well, first, the wording is not right. The inappropriateness, that comes directly from Abercrombie. He was infuriated that people would use contact inhibition to describe proliferation. He hated that. And there are heated debates at the time where people would argue one with the other just because of this wording. There is no common mechanism between the both. So you can add other sentences. That doesn't bring much to the but if you look now, this is not from us. I took it from this paper here, Pulifiato. The first letters are missing. If you look at the division time, or the cell density on the surface, you see that it increases over times longer than what you could see on the decrease of the velocity, right? So again, the two phenomena are very different and don't participate to the same thing. So in the following, I will address these two aspects. And the first one will be migration. What happens when you confine a monolayer and you release the barrier that constrains it into a spatial area? So you know the answer because Vincent told you about, but I'm going very fast on these notions again to reach more detailed studies we made on the leader cell that you've seen Saturday. So you remember that's how we work with this thing. The reason is, so we put a microfabricated stencil on the surface, place the cells into the apertures of these micro devices, let them grow to confluency or to the density we want, and once they reach this state, we remove the stencil, and that gives them some free space to migrate on. So this has many advantages. The classical test here would be to grow a monolayer and scratch it with a pipette cone. That's how people do it, and I've been doing it for years, or tens of years. The reason why we didn't do that and we chose this assay is because of several points. First, is it obvious that something will happen? So you know the answer because of Vincent, who destroyed my suspense. But is it obvious that something will happen, or do you need to somehow stress the border cell to trigger migration? So you know that you don't need that, but that was the first way to answer it. We don't affect the cell, we just release a constraint and they still migrate on the surface. The other thing is, with this assay, we don't kill or permeabilize any cell, and we maintain the chemistry of the surface in between the migrating epithelium. Okay, so here is what happened. So you remember this picture of a single cell migrating on the surface. Obviously, when it's surrounded by neighbors, even though it can develop lamellipodia under the others, it won't be able to move quite the same way. This is obvious, right? Because it holds the next cell, and the next cell holds the next cell, etc. So every time one cell needs to move, it will need to bring with it all the other cells with it. Well, some other cells with it. So obviously, this is the origin of correlated movement. So if you look at what happened in between, just after we remove the stencil, this is what you have. And you can see already with your bare eye that you have some movement in the monolayer. And since you can see it, it's because it's not diffusion-like. It's not like a diffusive movement. It's because it implies many cells together. Otherwise, you wouldn't be able, you would just see a blurry image. So there is this movement that is gone because I'm talking too much. And then you have this fingering that you see very clearly here that helps as a motor to bring the monolayer migrating on the surface. So these are the two main features that you can see. Maybe I can show you the first part again, so you can see. Here it is. So you can see it's moving very fast. Almost immediately after we remove the stencil, you have this movement within the monolayer. That we can map by PIV, so I don't come back to that. We can establish a velocity field at every time point in these populations by correlation techniques. And from which we deduce that, so these are the cells I'm dealing with mostly and that you've seen in the movies as well. They're MDCK. They're derived from kidney, and they're very strongly adhesive one with the other, right? Yes?
Can't it be a first step of spreading before migration?
Yeah, so there is that. The signal they get is that. The first layer just spreads a bit, and that is the signal that the other receive. Yeah. And so these MDCK cells, that's the velocity correlation function, the spatial correlation function. And what you can see is that it decreases over a distance of typically 10 cell diameters. So a huge distance, meaning that when a cell moves at some point, you have to go as far as 10 cell diameters to be independent of this first movement. A huge distance at this scale. Other cells, like fibroblast-like cells, NRK, whatever the name is, show they adhere much less one with the other. You can see some of them escaping from the monolayer. And when you measure exactly the same quantity, you see that after 2 cell diameters, you're already gone. You're already independent, much more independent cells. So I won't come back, on this V-check-like model, just to tell you again that this term is the major term that you add to your equation to witness this coordination between cells. In short, it says that if a cell is surrounded by its neighbors, we give it to this cell in the model with a certain weight, the average velocity of the neighbors. No more than that. And as Vincent told you, this describes very well what happened within the monolayer. Both, in terms of visual resemblance, but more quantitatively in terms of the spatial correlation function, the positions of the cell, etc. So I won't come back to that. Vincent told you all about it. But what this model doesn't describe, just with these ingredients, is the onset of these migration fingers. And you have them again here. While the movie goes, I can comment on these pictures. They're very long, very elongated. They're preceded by a cell that is different from the others. You can see it here. Their cells are larger. They develop a lamellipodium on the surface. These cells don't do that usually. They're epithelial cells. They don't make lamellipodia that easily. And much longer than the other. And the reason why we're so interested in them is because with this simple assay, we recover pluricellular structures like you have here. We see that emerging from the migration of the population, structures similar to what you have in 3D in other systems, like here, a multicellular protrusion stemming from a tumor explant implanted in a collagen gel. So these guys, the group of Peter Friedel, they've taken small tumor explant, put it in gels, observe what happened, and what they expected, I guess, I'm rephrasing the story, they're not here, is that they would see cells going out of this tumor like you have in the common epithelial mesenchymal transition. You go from a pluricellular tumor, and then you have single cells moving away, and these cells go away, they colonize other part of the systems, and then make other tumors, etc. And that's not what happened. See, what happened is that first, before all that, these tumors, they develop these pluricellular finger-like structures. And you have that in a variety of systems, actually. So these are, again, tumors extending protrusions in different situations. I think here it's on vessels, lymph vessels, on which they find also nutrients, so it's a bit different. Here it's the development of the lateral line in the zebrafish. Again, Vincent showed you this story. And actually, there is a huge range of different modes of migration from very individual to completely collective in all this part of this diagram that these people have identified. And so the idea that we have when dealing with cancer situation, that you go really from primary tumor to cells exiting from it and invading the medium, is completely, it's not right. You have a whole continuum of situations in between, illustrated by these different situations. This one in particular gives rise then to the so-called EMT, where cells scatter from this collective structure and then does exactly what I just described, goes in other places and seeds new tumors. So the resemblance here with these migration fingers that we experimentally get in our simple assay is then striking. You get very elongated structure preceded by a cell different from the others again. These fingers, these migration fingers, are lined with multicellular cable, actin and myosin cable. We'll come back to that in a minute. And develop a lamellipodium at the front end of this cell here, which is this leader cell I was mentioning. These are the cadherins, the protein that ensure the cell-cell adhesions. And you can see that there is also strong cadherin interaction between the leader cell and the followers. And last, if you look here at the adhesion between the cells and the surface, you can see that they're very localized at this leader cell. What else? Well, we can measure in this structure, in this finger, the velocity field and see how the velocity evolves from that point to that point and how the density evolves from that point to that point. And we can see that they follow very parallel paths. That the distance to the tip in many different fingers, and that the density or the inverse of the density is at the velocity. And now, if we plot all our data, including some that we can find away from these fingers, we find a very strong relationship between velocity and density. Yes?
Do you know why they migrate in these finger structures and not just as a whole front?
So why is always a tricky question. I haven't asked them. No, why I'm not sure. They do it. Maybe I can rephrase your question. Why is this leader cell different from the others to start with? That would explain it. Or is it just the emergence of something new? Would that? So this I will answer in the next slide. This I know the answer. Sorry. And so if we now put manually, sort of related to what you asked, if we put manually leader cells that are faster at the tip here, right, and integrate that there is something like an easy path when you follow this cell, so that two things help, the cable, and you've seen also the release of matrix by the leaders. And then this relationship between velocity and density, then we have more ingredients that we can add to the equation, and we can recover, as Vincent showed, fingers that look like the experimental ones. Yes?
Can you comment on the fact that they are often denucleated because
so, yeah, so that's, yeah, you're right. They're often binucleated. So that means that they divide, but the cell they don't go into undergo fission. I don't know why.
But as a consequence, as a transcriptional level, for instance, they're very different from all the neighbors. And for instance, in concert, you have many aneuploid cells.
Right. So the question is, yeah, right. So you're right. But it's a question of whether or not they don't divide because they're leader, or they become leaders because they don't divide. Every evidence we have is the other way around. We haven't seen any strong relationship between the cycle and the way they become leaders. But it's true that once they take this function, they don't divide anymore, for a reason that I don't know. The way I say it usually that they can do many things at the same time. But I mean, that's...
They still replicate.
They still replicate, yeah. Yeah, they can be trinucleated in some instances, or quadrinucleated, yeah. So yeah, so the question, so why maybe? Because they pull faster on the monolayer. So maybe there's an idea that it's better for the epithelium to cover as much ground as possible, even imperfectly, leaving some voids within, than just to grow slower on the surface and cover it perfectly, but in a smaller, larger amount of time. How does this stem? So several ideas have been tried. Some of them rely on chemotaxis. We haven't found any evidence of that, like the cell at the tip would emit some substances that would attract the others that would do it, but we haven't found any evidence for that. One explanation I like is due to Nirgov, who has introduced a coupling between curvature and the force or the velocity of the leading cell. So if you have that, obviously that will work. If you have some curvature, the cell at the tip will go faster, make even better, higher curvature, and then grow again, etc. So it's an instability. And that works well, at least at the very early stages. And then we assist to this change of phenotype of the leader cell. So the way we see it is that we have this small fluctuation that you will have anyway, that is amplified by this instability, right? And then there is a change in phenotype in the leading cell. We can go in the details of the cells constituting these fingers and try to make some mechanics to get some mechanical laws out of it. For instance, we can measure the cell orientation within these fingers. And what we found, so here, this line is what is a reference. It's what happened in the epithelium. And here, it's what happened in the fingers, in the migration fingers. So cells are much more oriented in the migration fingers, along the direction of the finger, as sort of expected. And the division axis is even better oriented. Cells divide in the migration finger with no problem, and they divide strictly in that direction, right? So this is exactly what you have, by the way, in this experiment. Exactly the same thing. You also have stretching and division along the axis. Although here you're in 3D, and here we're in 2D. What about the polarization? So this is different. Not only stretching, but how do cells polarize? So they polarize because they have a velocity, of course, that's obvious, and they all go in the same direction. They all go in the direction of the fingers. There's no ambiguity here. But you have other aspects that you can measure. You can measure an intrinsic cell polarity. And the way you do that is you measure the position of an organelle called the centrosome, or the microtubule organizing center, versus the nucleus. And what we observe is that this nucleus, in red here, mTOC, always point on average toward the leading cell, which is sort of not quite what you have usually. The usual law is that, as you can see here, away from the finger, is that the centrosome is between the nucleus and free space. That's what you observe in most of the cases. But here, it points toward the leading cell on average, with when they belong to the free edge, a small angle, witnessing that they also tend to go toward the free space. So it is balanced between these two contributions.
Can you somehow estimate the correlation distance between the leader and the rest, meaning that whether the cell knows how many cells are following?
Yeah, we'll see that. So these two measurements of orientation and polarization tell us that first, the leader cells seem to exert a force on the wall structure, a mechanical force, stretching the cells, etc. And second, that the cells behind it are still under active migration. They're not passively tracted by the leader cell, at least not only. So we can probe that. The way we can probe that is to stop the leader cell. And we have ways to affect it. We use a laser, and we sort of burn the actin in the lamellipodium of the leader cell. Doesn't kill it. Cell is still okay. At least it looks okay. No, it can recover in some cases, so it looks okay, really. But then it stops dead. Boom. It's like, the best way we found to pin it down. And indeed, if you look after 60 minutes, an hour, you see that the position is exactly the same, but things have changed behind it. Here we're very highly oriented at the velocity field, and not very dense in terms of cellular density. And here, the density of the cells have increased a lot, and the orientation is completely lost, right? So keep this in mind. It's very close to that, right? In both terms. So can we look a little closer to that? Yes, we can. The thing here is when we burn the actin, and what happened, what I haven't told you, is that what happened after some time is that a new leader starts and bring a new structure. It can be the same one as before, a new one, but you get to see a new structure emerging from that. And that's the green line. So what happened is that between the time when we kill the actin and the time where a new leader arrives, the cell density increases. That's just what we saw. And then when a new leader arrives, it decreases again. Okay. And that's the orientation of the cells. It decreases, and then it increases at a new leader. Right? Now, if we look at the velocities, we have the same shape of the curve. It decreases, so the other parameter related to the velocity, it decreases, and then increases again, as expected. Only what you see here is you have a shift. what happened on the velocities occurs before this emergence of a new leader. So the leader is necessary to bring the structure with it. Without it, if you kill it, then everything stops. But it's also the result of a prior orientation of the velocities behind it. The followers, in a certain way, elect the leader. They push it in front. What if we cut the finger in the middle? So we can even do better. We can cut it at different places, and I think that answers your question. What happens is that the finger retracts on both sides. And that's the distance over which it retracts. The closer you get to the epithelium itself, the less it retracts. So there is some kind of a complex tension field in this finger. But we can play around with that. And if we cut sufficiently close to the leader cell, like we do here, we cut it here. That's the first thing. What happens? So what do you think will happen? I'd like to do that. What do you think will happen for those who haven't seen that movie 10 times already? Try. What then?
It stops. It keeps going.
So you're both right. I love that. You're both right. It doesn't go anywhere, but it doesn't stop moving either. It doesn't know where to go. It needs a load to go, to have this persistent movement to bring the rest of it together. And see the other behind, they'll catch up, and as soon as they catch up, the finger goes again. Right? It's pretty impressive. Huh? I like this one. So it doesn't know where to go, but it's still there, and if you compute the velocity, the instant velocity, it's comparable to what it had before, but then as soon as it has a load, as a locomotive gets some wagons, then it can go. Okay, so the conclusion of all that is that the cells behind orient and elect a leader if you want, that then starts exert a force, and then altogether this will make a complex tension field in the finger, yes? We haven't done that. I mean, a few, between 5 and 10. No, I can't tell, between 5 and 10. Yeah. Yes?
And how does the wrist of the finger catch up?
Yeah, so they're still mobile. You have to see them as, going as much as they can, and on top of that, they're pulled by the leader cell. That's the way it works. We can test that, actually. We can test this sort of, at least this point. How does the leader exert a force on the rest? And that we can measure. We have tools for measuring that. You've seen some of these tools on the work on the bacteria by, I can't remember who. You. Which relies, traction force microscopy, which relies on bead embedded in a polyacrylamide gel, yes?
I've just been curious, has anybody tried to do DNA sequencing for the leader versus the...
So we would love to do that. We haven't done it yet. Maybe we'll do sometime. Yeah, that would be the right thing to do, I agree. Yeah. This is another technique to measure the forces. All these pillars are about 1 micron in size, in diameter, and a few microns in height. They're literally force sensors. They're silicon, they're made of silicon, and then when the cell pulls on them, it bends them, and by the bending, you measure the force. So we do that. The fingers grow, and they're very happy on this, at least on that picture. And they do what they do on flat surfaces, basically. And then we can measure the forces. So be careful. The scale goes from positive to negative. Positive is red, yellow. Negative is blue. So what you have, and this is a projection of the forces along this axis, along the axis of the finger. And that's the projection of the forces transverse to the axis of the finger. What you have here is that at the tip, you have very strong forces. That's unambiguous. And then behind, you have a succession of red and blue, and it's very, I mean, the main thing is it's very noisy, right? It doesn't appear to be that obvious what it does. It's very noisy. And here you have more blue here and more red there, so it's easier. You can see immediately that you have forces on the sides that are centripetal. They tend to converge toward the center of the structure in that case. Here, it pulls here, and what happened behind is less obvious. So we can make profiles over many of these fingers, and when we make that, we see that on the transverse side, we recover that these forces, positive and negative, toward the center. And here, we have this positive force here, meaning that the leader cell grabs the surface, pulls on it, brings the other with it. And then, along this profile, the curve gets inverted. It becomes negative on average. Locally, it can be anything. Locally, it's anything because the cells precisely are migrating, so they exert forces in all directions. But if you average out everything, then you get a friction behind. And this friction you get behind, you get this curve, and this curve immediately made us think that it looks like a single cell. A single cell is a dipole of force. With positive force, well, a dipole. Force is converging toward the center. And what we have here is exactly that. So we have what people call a supercell. I was very dubious, I must tell, at the beginning using that word. But it's actually pretty accurate. You have to realize that we get, at this scale of, let's say, 50 cells, between 30 and 80 in the largest we had, the same force dipole at a completely different scale that we have for a single cell. So the whole tissue reproduces the individual behavior of each cell. So that's a pretty important conclusion for that. Maybe I'll skip that one. How much time do I have? This is what happened on the sides. This is a cable. Well, this is important because this is a cable that you have, this actomyosin cable. This is what acts as like a muscle along the edges of these fingers. You have it here very well, and here also you see the myosin-like contractility that forces the cell to grab the surface and pulls on it. And this cable is under tension, as you can see. If we cut it with a laser, then it retracts on both sides. Yes?
What is the kind of interaction between cells? I mean, what keeps the two cells close to each other?
So they link one to the other via a protein called cadherins. And this cell type in particular is rich in these cadherins. So they really are bound one to the other. So this is good and well, but can we go a bit further and go beyond the mechanics of these structures? And one way to do that is to address this problem is to say, how is this mechanical? Can we have an integrated picture where we would have the mechanics of these fingers that we just got with the activity of the proteins involved in kind of systems? And one of the good candidates for that is RhoA. RhoA is a GTPase that basically, if I want to make it short, control the actomyosin contractility. And so if we can find a way to measure in parallel forces and activity of RhoA, then we have a good vision of what happened regarding contractility on both aspects of biochemistry and mechanics. And so the first thing is if we put inhibitors, indeed, we get inhibitors of RhoA or siRNA against RhoA, we get much less fingers, less developed, less active, etc. I mean, already that's the first thing we can measure. But we can use better tools for that. And you have to realize here that we don't want to measure the concentration of these proteins because you have plenty of it everywhere. It's a very abundant protein. What you need to know is the activity of that protein at certain places and in time zone space, if you want, the dynamic activity of the protein. And for that we have tools, and this is one of these tools that we have used with Jacques Camonis next door. It's a FRET biosensor. And this FRET biosensor you can transfect the cells with, and when the protein RHO is active, the conformation changes, and the two labels you have on this protein then emit a FRET signal. So they're not that easy to work with, I tell you the truth, but still you can use that just to measure locally the activity of this protein. So we've done that, and if we do that, what you have, this is a live image. doesn't move, but it was taken on live cells. What you have, if you look at the fingers at the leader cell, you see that you have more activity at the front end compared to the back end. Okay? Now, if you look at the cell within the finger closer to the epithelium, you have the opposite distribution. More activity at the back of the cell compared to its front. The tip is over there. And if you look at the cell belonging to the edges, where you remember you have this actomyosin cable, which is here, then you have more activity facing the free surface than facing the other cells. Can we try to be more quantitative than these pictures? Yes, we can measure for each cell what we call the differential activity, which is basically the gradient of activity front, back, or left, right. And if we plot this differential activity versus the distance, this is the tip cell, and this is the epithelium. Again, for many different fingers, we integrate all these results, etc. This is what we have. Positive at the front end, at the leader cell, as you saw, and negative closer to the epithelium. And that's the distribution in the transverse direction. And again, what that just means is that it has a higher activity when facing the free surface compared to the other cells. And of course, when you see that, just a second, when we see that, the first image we have is that it's exactly like the force profile we had before. So there is perfect correlation between the force profile and the RhoA activity profile. Very different experiments, but they just show the same thing by two different channels. Yes, Lisa?
How do I reconcile that plot on the left with the observation that the centrosome is organized to the front tip of cells?
So there are still open questions. That's one of them. I don't know. I don't know about that. The literature, as you know, is full of, observations and then counter-observation. I would have expected what you say. That's not what we have. Yes?
So people also work on similar kinds of experiments in 3D tissue models, like suspension cultures?
Probably. Probably. I don't know all these details, but probably, yeah. So the main difference, I won't answer your question because I don't know, but I will answer another one. When we show that, and we have this analogy with Peter Friedel stuff and migration finger, etc., everyone says, oh, whoa, it's very different because in these 3D migration fingers, the main thing you have, and that's true, are collagenases that eat up the collagen gel, and then it makes things easier for the cells behind, and they all go there and it goes there. Even the leader cell can even be a fibroblast in some cases. Okay, right, understood. I agree. But here, it's actually not that different in the concept also, because this thing here, actually what this does in 3D, it makes an easy path, right? And once the leader cell here has gone and developed this cable on the sides, it makes actually a similar kind of thing. It makes an easier path for the cells to follow that and not go to the rest of the medium. And I have a proof of that. Okay, so let me continue that, and I'll come back to that in a minute. So that, of course, you can plot one with the other. This is force, and this is the differential activity. You can even measure it in the same experiment, same time, same cells. You measure both the force and the activity of the protein, and that's a challenge that the postdoc at that time, Myriam RhoA, was able to do. And you have a good correlation with much scatter, obviously, but you have the same correlation that you had when average out pulling many experiments together. So this is what I wanted to say. If you break this cable, and we can do that because I've shown you an experiment where we did. So we cut the finger with the laser here. That's how we see that it's tensile, it's contractile. Now, what happened after is that, in many cases, not all, but in many cases, see, there is a finger developing here. So actually, this muscle that you have on the side, quote, muscle that you have on the side, prevents other cells to move on the free surface. So it acts as a gel in the 3D case you mentioned. And that's why you have long fingers. That's because as soon as you pull on the structure, you develop this cable that itself reinforces via this force signaling, and that prevents other cells to go and branch on it. So the cable here is very important for that reason. The other aspect is that if you play the movie backwards, so you know which cell will become a leader cell, and you play it backwards, so you start here and you go there, that's the leader cell before it's anything. And what you can see is that the activity of RhoA is completely homogeneous on that cell, contrary to here where it's polarized. So it's not predetermined in that respect as well. It's not a cell which is already polarized in the right direction that will bring out the population behind it. Okay, yeah?
Where does this actin cable come from? Is it there before?
Okay, so no. It develops, so it's a very ubiquitous structure. You see that all the time for this kind of cell, epithelial cells, as soon as you have a border. And the way you should see it is that it's like an actin cortex, very well developed at that point, and it is not continuous per se, but it does mechanical relays that are the cadherins we were talking about before. So each cell has its own small cable, and all these cables are stuck together via the cadherin. So it makes us effectively pluricellular structure.
And when the two tissues finally merges, what happens to the cable?
So the cable disappears.
Okay.
Yeah, eventually the cable disappears. The leader cell, it depends a bit. If it's already binucleated, it has more trouble to go into the crowd. But other than that, it's, yeah.
How flexible is actin?
How?
Flexible? I mean, how bendable?
Yes. Is what? The cable?
Yeah.
I don't know. I mean, it's a bundle of many actin filaments. I can't enter that. Doesn't appear to be that flexible, but that's all I can tell. Okay, so this summarizes most of what I said, and outline the importance of the cable because it's, as I said, ubiquitous. You see that all the time. And here its role is really to prevent the emergence of new leader cells on the side. What happened before releasing the constraint? And there, again, I'm going to take it on what Vincent left. So you remember we grow cells in these patches. So that's about the equivalent of saying what happened before we peeled off the stencil. It's the same kind of experiment. So we have these patches of MDCK cells. I can show you the movie again, maybe. And you can see that there are two different stages. At the beginning, cells move a lot. You can see that, right? You still have a lot of movement. And then after 30 hours, it's become very quiet. And you have the appearance of these structures on the side and in the center, right? So it's very lively here, move a lot, wiggle a lot. And then it becomes quiet and new things happen, let's say. Okay, so what happened at the beginning is what Vincent told you. You have these oscillations, both in rotation, but not really oscillations, stochastic change of direction of the solid body that the cell make. And on the radial direction, you have this breathing oscillation that indeed you can describe, with exactly the same model that he developed for the migration of cells. So I'm not sure he has emphasized that enough. We don't touch the parameters. We use exactly the parameters that we got from the migration experiment. We plug them in the confined situation. We just use different boundary conditions. And what happened is that we recover the existence of these oscillations plus a quantitative agreement in their period versus the size of the domain. So that was good. What happened at long times? You saw that the situation was very different. And that's going to address the second point I had, which is contact inhibition is different in proliferation and in locomotion. So that's what happened in a normal epithelium. Look at this one here. If you wait for long enough times, you see this whitish deposit in the center of the domains. We have that all the time. And what this is a result of extrusion of cells from the monolayer once the density becomes large enough. So cells grow, divide, the density increases, and at a certain point, they don't want more people there, so they just expel them. It's a very easy way and very fast response to overpopulation, if you want, to overcrowding, because it doesn't need any regulation of proliferation per se. It's completely passive. Too much cells, then they're going to be expelled away from the monolayer. And we have that all the time. So it's called extrusion most of the time, or delamination. We can find that. There is debate here, an issue whether or not these cells are alive. The consensus now seems to tend whether they're still alive when they're expelled, and then they die or not, or I don't know. But basically, that's what happened at the beginning. However, if we look on our pages, yes?
What decides whether a cell is expelled from the monolayer or not?
Sorry?
What property decides whether a certain cell is expelled from the monolayer or not?
Which cell? Well, that's at the division point. When there is a division, again, the details, I'm not completely sure, but one of the daughter cells is expelled from the monolayer. That's apparently randomly. But that's not quite what we have. If we look at the border where we see this structure here, you see this is phase contrast, and if you can see this white halo, it means that there is a change in curvature. And this kind of curvature corresponds to this in fluorescence, that's a confocal stack. And what you have is a rim at the edge of this monolayer. So the border itself gives rise to new structures that are different from what you have in the center. Just because cells are at the border and not in the center of the domain. This is even better here. And this rim that you have at the edge doesn't depend on the domain size itself this time. It's completely determined by cells at any kind of border, any kind of edge. You can see the width doesn't depend on anything. And if you take a too small domain to start with, the two edges fuse in a single one, as you have here. It makes a little droplet. So why is it interesting? It's interesting because the dogma in these cell systems is that you need to adhere to a surface to keep the cells alive. They need that. And that's what happened everywhere here. They don't go that much away, and that's why there is this debate when cells are extruded, are they alive or not, etc., and how long. But what you have here is that cell-cell contacts can compensate for that. This cell here obviously hasn't any interaction with the surface, but the interaction it has with its neighbors is enough to stabilize it and keep it alive. The other thing is, if you look at the polarity, whatever the protein here, it's aserine, it goes to the apical side, you can see that the polarity is defined at the pluricellular scale, not at the unicellular scale, like here. The whole thing is polarized. So where does this come from? Well, this comes from not from a flux of cells from the inside to the outside, as we could have thought at first, but mostly, if not only, by proliferation. If you look at what happened at late times, you see that you have this single cell extrusion in the center, but almost nothing here in this area which will become the rim. And the reason for that is that just quantifies it. And the reason for that is that in the center, we have what I just described, cells trying to divide. They can't. They expel one of the daughter cells into the third dimension where it stays or dies, or that's what happened. But on the edge, that's not quite what happened. If you have a cell, so you start from this mother cell here, that's divided there. One of the cells go to the floor, to the basal side, if you want, while the other is incorporated into this rim. So the edge makes the cell to incorporate extra cells and make a new kind of tissue rather than expelling them away in the third dimension as it is the case in 2 dimensions, just because you have an edge. Which, by the way, may look like the precursor of maybe an organ. These cells, they are derived from kidney. When in the right condition in 3D, they can make tubes, like we have in our kidneys, tubules. When they're stimulated with growth factors, they do that also. And here we have nothing of the sort, but they do it spontaneously just because of constraint. They develop this 3D structure, which may be the first step of a tubule. Maybe. We haven't seen tubule exactly, but we see these are called cell cords. They're precursor to tubules. OK, maybe in the next 5–10 minutes, I'll show you a different kind of consequence of confinement. And these are newer work that we have on cells that don't adhere one with the other. And these cells are these fibroblasts. They're very elongated, as you can see. And when they migrate on the surface, they basically show no polar movement. They don't go, from one place to the other, as many of what you've seen. They don't exhibit anything close to Brownian motion. So they're sort of active rods moving on the surface, as far as we can tell. They're apolar. And they don't have adhesion one with the other in that case. And then when they come to confluency, when they grow and grow, this is what happened. And you can see directly that you have some long-range orientational orders. You can see it, right? Yes? OK. And so that was interesting. Why do they do that, and how can they cope with that, and how can we study that? Is it interesting? So first, they are completely out of equilibrium. If we measure the density, the local density, and its fluctuations, it's a signature that this is an out of equilibrium system. That's to start with. It's completely active. And then when we try to quantify the orientation of the cells in the monolayer, this is what we have. So this is phase contrast. Here we have just color-coded the various orientations we have in the monolayer. Like this one would be a data angle, etc. And you can see that locally they're extremely well oriented. And then these patches of good orientation are somewhat juxtaposed one with the other. And here you have the orientation field directly. This is the contact inhibition that we had before. This we keep. And we can quantify the amount of order by this order parameter that you have here. That doesn't preclude any direction, because there is none in the system that we should refer to. And then if we look at the way it evolves, it grows until it's saturated to a certain value. And of course, depending on the window size we have, this certain value will be different. If we take something small, then we are going to be within one of these patches. And then we go to 1. If we take a very large one, we have many of these different orientations to take into account, and it will saturate to this value here, which is intermediate. All right? 1 is perfect order here, and 0 completely random. And the density increases at that time. So there are two effects that play. I'm going to back to that together. So if we measure the autocorrelation function, we get a certain length. We can do that at different times. And again, we get an increasing curve of this correlation length versus time, which saturates around 500 microns. So if we wait long enough, then we have domains of a typical size of 500 microns, within which cells are perfectly oriented. And these domains are juxtaposed one with the other. So that's the point I wanted to make. As time goes, two effects happen. The first one is the contact inhibition of locomotion I mentioned before. Movements progressively freeze. Everything is going, it's more and more difficult. People have modeled that in terms of jamming or other kind of transition. But basically, that's what happened. Everything gets more difficult, and everything just freezes. But also, as the density increases and cells are pushed one to the other, the order increases. So time has two consequences. It helps the order, and at the same time, it decreases the motor of this order, if you want, which is the intrinsic mobility of the cells. what can we, yes?
What sets the length scale of the clusters?
So what sets the length scale of the clusters? This I'm not sure. It's a good question, but that I'm not sure. It just we measure. That's what we measure, and we have no real. I don't know. I don't know about that. I think it's intrinsic to the cell type, their shape, the way they interact together, etc. So yeah.
These cells are only moving in one direction, right? Or are they also moving in both directions?
No, they move in both directions. That's the movie you had at the beginning. Here, see that they basically do that. So they're apolar, as much as we can tell. There is no preferred direction. And again, I think there are aspect ratio, the interaction between them, etc. All that comes to having a correlation length of that much. But I can't go back to the macroscopic details on that basis, not yet at least.
The order parameter is the global order parameter?
Yeah. So the other parameter I put here is over the field of view. But that's a notion I'm going to discuss in a minute. So when I show this to people who know what a crystal, liquid crystal is, which I'm not sure I know, the thing is, the thing they want to see are defects. The liquid crystal community, they're driven by the shape of the defects and how they look like on the microscope. And so I told them, yeah, you see we have cellular nematics. And they say, well, show me the defects. So we looked, and we found the defects. They're outlined here. And basically, we have the two types of characteristic defects of the nematics. The -1/2, which is here, and the +1/2, which has this comet-like shape. And these defects are precisely what prevents these domains to fuse. Because as time goes, again, everything freezes. So the defect, these two defects can annihilate when they're close enough one with the other. We've measured a few of these events. So they can annihilate, but when when they can't move really one to the other because of the high density and this contact inhibition of locomotion, they just stay where they are, and that prevents the domains to merge one with the other. The defects prevent having a macroscopic nematic field. But we can verify also on them that they go in the direction you expect. So this one you see as it's completely center of symmetry. You don't expect it to go anywhere. You don't expect it to move. And that's what you have in this red circle. It stays where it is. Doesn't move at all. This one here, on the contrary, has not the same symmetry, and it can move. It can move actually in both directions, from top to bottom, for extensile systems, which are the most common. And toward the tail of the comet, if you want, is the contractile systems. Now, if you look at our examples here, what you have, if you follow that one, you can see very clearly that it goes into that direction outlined by the arrow. So we have a contractile system, which is not that surprising given the way a cell moves on the surface. And that describes very well what the situation we have. So now, is there a way to come back to your question over the window size over which we measure the other parameter? Is there a way to get to completely oriented monolayers? What do you think? How would you do it? Pooling, or yeah, well, maybe. Think of what people would do or are doing with classical liquid crystals. How do you orient a liquid crystal? You can flow it. That would be not easy to do with cells, but that would be a possibility. Like a force, by the way. These are good possibilities, but it's harder to do. Yes? Yes, you can annihilate, but again, here we depend on the activity of the cells. So,
You put some food on one side?
yeah, right, okay, you can. I haven't thought of that one. I'm not sure these cells are chemotactic, by the way, but maybe. Yeah?
More density.
The density, we can't play that much on it, because if we increase the density, we freeze everything. So we can't really get to to the optimal situation. Boundaries, that's what I wanted to hear, boundaries. What people do in liquid crystals is that they rub the surface in a certain way so that the molecules anchor to this surface with a certain orientation, parallel or perpendicular. So we can induce boundaries here. That's actually, okay, pretty easy to do. We can pattern the surface. So that's what happened when we pattern the surface. And we try to grow these cells within these stripes. And what you're going to see here is that progressively they orient into the right or into the expected direction, right? Just by following the recipe that liquid crystal people told us. So you have the same thing here. The color code shows you that after 60 hours, you're completely oriented in that direction here. And you get, if you measure now the other parameter over this population here, you get a value of 1 at higher density.
What's the rate of cell proliferation?
The what?
The rate of cell proliferation.
Like all the cell, I get one. There are 20 or 24 hours between divisions.
So for the last time step here.
Yes, you increase the density with the proliferation.
Is there a transmission of the orientation?
Yeah, well, that goes in the right direction also. When they divide, they tend to keep the same axis. That's your question, right? Yeah.
We heard earlier in the course that with Chapter, I think it was, that people play with the little solid rod particles that they shake, and they can also self-orient and show thematic behavior.
Yeah.
Do you see qualitative difference?
Well, we see qualitative difference because of that, because we're contractile. And then the net output will be different in terms of defect annihilation, in terms of dynamics. I mean, it's the same framework, of course. Hugues, maybe you have a comment on that. But it's the same framework, but there are differences due to that, I think.
Yeah. Now, the main difference in what I showed is that here, obviously, density plays a role.
Right. Beside that.
And we have one part. Now, if we could give them a little bit of, the size, some repulsive potential, and crank up density up to similar levels, we do see effects. They do have the non-equilibrium dynamics, and the way they move, actually, the minus plus 1/2, the way it moves depends on parameters.
You can tune it.
Yes.
Yeah, definitely.
This gentleman here in front of me actually has performed very interesting simulations of hard rocks. So that's, the only interaction actually is explicit collisions between rods which are reversing the velocity at finite times, and he sees exactly that.
Yeah.
And that's no effective alignment that you could, it's
all
just
right.
colliding
Yeah.
on there.
Yeah.
This guy, that's just the right timeline. When you speak now, it's okay.
Okay, so how can we tune the widths of these stripes in order to get this parameter, the other parameter of 1? With not big surprise, we come to the correlation length we measured before. As long as we're below 500 microns, we get a perfect order in the stripes. If we get larger, then we let domains develop within. And as in liquid crystals, I'm wrapping it up now, it starts from the edges, from the boundaries. See, that's the alignment, and that's the time, and we integrate it over the whole stripe. And so they start by being well aligned on the edges. Density increases. You don't see it in this color code. I can show you the movie again. And basically, the order propagates from the edges, from the boundaries, to the center, as you see here. You have more and more red at the edges, and then it goes toward the center. It fluctuates a lot, but eventually, you get the perfect alignment within these stripes. Here it is. So we can play games with that. It's actually pretty fun. Let's take 2 branches, like in a cross, and we play around this length of 500 microns. So I think this one is always 400, and this one is the vertical one increases. So the horizontal one is always below, in principle, the correlation length of 500 microns. So what happens if you have, here you expect perfect order if they were not interacting in both cases. But since you have less cells in the vertical branch, what you have is that the horizontal branch always wins. This one is always at perfect order, and this one interrupts at these points. Just because you have less cells to compete with these ones here, and they all have the same orientational information, if you want. Here, you have 400 microns in both directions, and with no surprise, well, to our surprise, I should say, one of them wins all the time. Not always the same one, with a 50% probability this one or that one. But there is one continuous path, and the other interrupts. And then as we keep increasing this size here, we get less and less order, but you can still see that here, barely, but still, you still have a continuity of the order in that direction, which is close to being perfectly ordered, because we have, again, more cells into this branch compared to that branch. And here, you're in complete mess. OK, so well, basically, the student who did that, Guillaume Duclos, likes to say the group size affects the propagation of information. It's a fancy way of saying what I just described. OK, so these are the people who contributed to these different experiments that I showed you. Myriam Refait performed the work on the fingers, on the migration fingers. Maxime did most of the work on the confined epithelial domain. Guillaume Duclos, on the nematic cells, and we've collaborated with all these people. Thank you very much.
So we're going to do a little coffee break, but we have time for 2 questions before we go for it. So is there any question? Right. So maybe I have a little question actually. You presented this contact inhibition of locomotion. Actually, it was RhoA that activates the migration into the other side. And with this finger, it's the opposite, like RhoA seems to be completely activated on the other side of the cell. And so how you can reconcile the fact that you have completely different signaling dynamics within the cell?
So I think the finger and the leader cells are completely controlled by contractility. They're not migrating as single cells. That's your question, right? Single cells have this signaling in a different way. Here, I think that contractility is much enhanced and the role of contractility compared to the migration of single cells that goes with lamellipodia, etc. So I think part of the answer is there, but part of it is we're still surprised of that, and there is more to do. Yeah, I'm not completely sure of that either.
And the other question is about how this alignment, how you think that could be set within the formation of a body, for example. If you want to align in many cells, there examples in organs.
So there are many of them deal with neurons. Very often in nerve cells, they are perfect order. What maybe I should emphasize here is that usually you find many papers that tell you that if you want to order cells, you have to nano-pattern or micro-pattern the substrate to make ridges on which cells will grow and crawl and make a very... And here, what we've seen is that it's not quite necessary. You can make a structure much larger than a single cell and still have a very good alignment of the whole population. So that's a side remark. Basically, most of you can find deals with mixed population of cells, fibroblasts in some cases, mostly nerve cells. That's where you can find most of it in vivo. Yeah.
Okay, so you guys have no questions. Let's have a coffee break. So maybe